This procedure begins with removing the cerebellum from P4 to P5 newborn mice. Meninges and the choroid plexus are removed from individual cerebella, which are then collected in a 15 milliliter conical tube. The cerebella are dissociated with papain and brought to a single-cell suspension with gentle trituration. This suspension is filtered by gravity through a nylon mesh with a 70-micron pore size. The flow-through is gently layered on top of a discontinuous Percoll gradient and centrifuged to separate the granule cells from the glia, interneurons, and Purkinje neurons. Cells in the interface of the gradient are collected and pre-plated on poly-D-lysine-coated plates to further remove the glial cells. Granule cells are collected, counted, and plated at desired densities on poly-D-lysine-coated glass coverslips or plastic culture plates.
Hi, I'm Hae-Young Lee from the laboratory of Dr. Lloyd Green in the Department of Pathology at Columbia University. Today we'll show you a procedure for isolating and culturing cerebellar granule neurons from postnatal mice. We use this procedure in our laboratory to study granule neuron progenitor, proliferation, and differentiation. So let's get started.
This procedure begins with using 70% ethanol in the dissection area. To dissociate cerebellar granule cells, we use the papain dissociation system. When starting a new kit, prepare the albumin ovomucoid inhibitor solution. To do this, take 32 milliliters of Earl's Balanced Salt Solution, or EBSS, which is provided in the kit, and add it to the albumin ovomucoid mixture. Set it aside while preparing the other components to allow the contents to dissolve. Next, add 5 milliliters of EBSS to one papain vial from the dissociation kit. Each vial is sufficient to dissociate 4 to 15 cerebella from P5 mice. Then place the papain vial in a 5% CO2 37 degrees Celsius incubator for 5 to 10 minutes until the papain is completely dissolved and the solution appears clear. Maintain the solution at room temperature during dissection. Next, add 500 microliters of EBSS to one DNase vial from the kit. Mix gently by tapping the vial since DNase is sensitive to shear denaturation. Add 250 microliters of this solution to the vial of papain to obtain a final concentration of 20 units per ml and 0.005% DNase. Save the remaining DNase vial for use in later steps. Once the tools and reagents are ready, proceed with dissecting the cerebella and removing the meninges. For this procedure, C57 black 6 mice are used between postnatal days 4 and 6. This age range is optimal for granule cell culture because the number of external granule layer or EGL granule cell progenitors is at a maximum. To begin, pipette 15 milliliters of HBSS glucose into a 60 millimeter plastic tissue culture dish and 10 milliliters into a sterile 15 milliliter conical tube. Place the tube on ice. Wipe the head of the pup with 70% ethanol. Use permaset scissors to remove the head. Transfer the head into the hood and hold it with forceps so the back of the skull is clearly visible. Insert the microdissecting scissors into the foramen magnum in order to access the brain and then cut straight toward the eyes. Use fine Dumont No. 5 forceps to peel away the skin and lift up the skull to expose the brain. Then pinch off the cerebellum and the surrounding midbrain. Transfer it to the dish containing HBSS glucose. Note that it is easier to manipulate the cerebellum and to remove the meninges if the cerebellum is still attached to the surrounding tissue. Next, place the tissue under a dissecting microscope. Then use one set of forceps to anchor down the cerebellum to the plate. Use fine Dumont No. 5 forceps to gently peel the meninges off the cerebellum as well as between the lobes. You will notice blood vessels on the surface of the cerebellum. These blood vessels are a good way to identify the meninges. Continue to remove the meninges until the cerebellum takes on a matted white appearance. Then separate the cerebellum from the rest of the midbrain. Turn it on its ventral side and remove the choroid plexus, which looks like a reddish ribbon between the ventral cerebellum and the adjacent midbrain. As soon as the cerebellum is dissected, place it in cold HBSS glucose in the 15 milliliter conical tube on ice. After dissecting 3 brains, replace the dissection solution with fresh HBSS glucose. As you become more familiar with cerebellar dissection, try to reduce the dissection time to no more than 6 minutes per pup. When the dissections are complete, proceed with the cell suspension step. To generate a cell suspension from the cerebellar dissections, first remove the HBSS glucose from the tube and replace it with papain solution. Although the kit recommends cutting the tissue into small pieces, we find that it is not necessary at this age. Place the tissue and papain solution in a 5% CO2 37-degree incubator for 15 to 20 minutes. For 4 to 8 cerebella, incubate for 15 minutes. For a larger number, incubate for up to half an hour. Gently agitate the tube every 3 to 4 minutes. Once the incubation is complete, begin the trituration step. Coat a sterile P1000 aerosol pipette tip with FBS by pipetting up and down twice. Now triturate the mixture. Make sure to pipette gently to prevent any air bubbles from forming. About 10 up and down movements with the pipette are enough to dissociate the tissue. The solution should become cloudy. Allow the suspension to sit for 30 to 60 seconds so that any large pieces of undissociated tissue will settle to the bottom of the tube. Although fire-polished glass pipettes can be used for trituration, we find that serum-coated sterile P1000 tips work just as well. Next, remove the suspended cells into a fresh 15 ml conical tube using a serum-coated pipette tip. Be careful not to remove the undissociated tissue pieces at the bottom. Then centrifuge the cell suspension at approximately 200 G for 5 minutes at room temperature. During the spin, prepare the resuspension medium. To do this, add 2.7 milliliters of EBSS in a sterile tube. Then add 300 microliters of the reconstituted albumin ovomucoid inhibitor solution. Finally, add 150 microliters of the DNase solution. Once the centrifugation is complete, discard the supernatant and immediately resuspend the cell pellet in the diluted DNase albumin inhibitor solution. Again, use a serum-coated pipette tip to resuspend the pellet. Now prepare a discontinuous density gradient. To do this, add 5 milliliters of the albumin ovomucoid inhibitor solution to a 15 milliliter conical tube. Layer the cell suspension on top of the solution by gently adding the cells along the side of the tube. Then centrifuge at approximately 70 G for 6 minutes at room temperature. The dissociated cells will pellet at the bottom of the tube while the membrane fragments remain at the interface. If you wish to obtain cultures that are enriched in granule cells, there is no need for further purification. Discard the supernatant and immediately resuspend the pellet in 4 milliliters of 10% FBS medium with 50 microliters of DNase. Filter this suspension by gravity through a nylon mesh with a pore size of 70 microns. This step removes large non-neuronal cells and provides for a better single-cell suspension. Then bypass the following Percoll gradient separation step and proceed to the cell plating step. Or, if you wish to further purify the granule cells from glia and large interneurons, proceed to the Percoll gradient step. Discard the supernatant and immediately resuspend the pellet in 2 milliliters of HBSS glucose with 50 microliters of DNase. Again, filter the suspension through a nylon mesh. Before beginning the Percoll gradient separation, make 2 fire-polished Pasteur pipettes. Gently flame the tip of a pipette until the edge is smooth and the pipette bore is about half of the original size. Be careful not to make the opening too small. Now make the Percoll gradient. Place 10 ml of 35% Percoll solution into a 50 ml sterile conical tube. Then load 10 mL of 60% Percoll into a 10 mL sterile syringe with a sterile spinal needle attached. Gently layer the 60% Percoll solution underneath the 35% solution by letting the syringe tip touch the bottom of the tube. Take care to keep a sharp interface between the two layers. Next, add the cells to the top of the gradient using a fire-polished pipette. Gently add the cells along the side of the tube without disturbing the interface. After layering the cells, carefully place the tube in the centrifuge. Start centrifuging at approximately 18 G. Ramp up the speed one step every 20 seconds so that it takes approximately 2 minutes to reach 1800 G. After 10 minutes at 1800 G, decrease the speed gradually one step every 20 seconds. When the spin is complete, remove the upper interface of the gradient and discard it. This interface contains large glia, Purkinje cells, and interneurons. Next, use a fire-polished pipette to carefully remove the cells at the interface. Transfer the cells to a 50 ml tube. If the volume happens to be greater than 15 mL, then split them into two 50 ml tubes and resuspend in 3 volumes of HbS's glucose and mix by inverting several times. Then centrifuge for 5 minutes at 1100 G. Now remove the supernatant and add 4 mL of medium containing 10% FBS and 50 microliters of DNase. Resuspend the pellet gently using a fire-polished pipette to form a single-cell suspension. Once the cells are resuspended, proceed with pre-plating the cells. Pre-plate the collected cells on a poly-D-lysine-coated dish for 20 minutes in an incubator at 37 degrees Celsius and 5% CO2. During the incubation, the astroglia and heavier cells will settle down and adhere to the dishes, while the small granule neurons and neuron progenitor cells will remain floating or loosely attached. After 20 minutes, gently tap on the dish to dislodge loosely adhered granule cells. Then transfer the cell suspension to a fresh poly-D-lysine-coated plate and repeat the incubation. Next, collect the granule neurons and neuron progenitor cells in a 15 ml conical tube and centrifuge at 200 G for 5 minutes. Then resuspend the pellet in 1 ml of serum-free medium and count using a hemocytometer. Finally, add serum-free medium to reach the desired cell density. To plate at medium density for 6-well plates, use approximately 3.5 to 4 million cells. For 4-well plates, use 650,000 cells. Maintain the cells in a 37-degree incubator with 5% CO2. Change the medium completely 24 hours after the initial plating, then every 2 to 3 days thereafter. Here are some images of healthy granule neuron and progenitor cell cultures 24 hours after plating and 6 days after plating. These cells can be maintained in culture for up to 2 weeks. Note that within 24 hours after plating, healthy cells will spread evenly around the coverslips or the plastic well and will project processes. Cell clumping and the presence of varicosities on the cell processes indicate that the cells are unhealthy or that the poly-D-lysine substrate is toxic. As seen in the diagram, granule neuron progenitors begin to differentiate upon plating and express markers representative of their differentiation state. The proliferation of granule neuron progenitors can be substantially prolonged by adding sonic hedgehog to the medium. Laminin can also be added as a substrate in addition to poly-D-lysine to promote neurite outgrowth. These features of the granule cell culture offer a system for studying either the biology of granule neurons or the regulation of progenitor cell differentiation into neurons.
We've just shown you how to isolate and culture mouse cerebellar granule neuron progenitors and neurons. When doing this procedure, it's important to remember the approximate yield is 4 to 5 times 10 to the 6 cells per pup, and plating densities can range between 1 and 5 times 10 to the 5th cells per centimeter square. So, if the Percoll gradient step is bypassed, then 8 postnatal day 5 mouse pups can yield enough cells for 6 4-well culture plates. In my hands, the use of the Percoll gradient step will reduce the yield of cells approximately 20 to 35%. However, the percentage of granule neurons in cultures increases from 90 to 95 to 99%. Enriched granule cells obtained without the Percoll gradient separation step have been used to study the regulation of granule neuron progenitor proliferation and differentiation. However, if the cultures are to be used beyond several days in vitro, inclusion of the Percoll gradient step is recommended in order to decrease the contamination of the granule neurons by proliferating non-neuronal cells. Also, make sure the solutions used for making the Percoll gradient is fresh, as improper buffering leads to greater cell death. So that's it. Thanks for watching and good luck with your experiments.